Hello and welcome to the 2021 Nobel Prize Lectures. This event is brought to you by Simon Fraser University's Faculty of Science. We hold these every year to celebrate the Nobel awardees and their impressive research work and to allow our community and friends to engage in meaningful conversations with our university faculty members about this research. I would also like to acknowledge that today, February 11th, is the International Day of Women and Girls in Science, an important day in recognizing that gender equality and the empowerment of women and girls is vital for the future of the sciences. Today, we recognize the contribution of the 2020 Nobel Prize awardees in chemistry, physics, and medicine and physiology. Experts from these fields will highlight the impact of these prizes and the connection between fundamental and applied research. My name is Dr. Corina Andreoiu from SFU's Department of Chemistry, and with me as co-host this evening is Stuart McNish from Oh Boy Productions. Hello, Stuart. Thank you for helping us with this event.
Well, it's my pleasure, Corina. I'm pleased and honored to be a part of this celebration of science and research and to be able to engage with our faculty presenters this evening. You know, it's great that Simon Fraser University stages this event every year, as it is a fitting opportunity for the general public to learn more about science research, the Nobel awardees, and their impact on our everyday life. I'm also excited to learn more about these contributions. I also want to extend a warm welcome and a hello to all of those joining us virtually this evening. We have an impressive number of enthusiastic viewers tuning in, and we thank you for your interest in the event this evening. To help keep this event interactive and allow you an opportunity to participate, we have a question and answer field on the right-hand side of your screen. And we encourage you to type in any question or comment you have about the discussion and presentations. We'll have time at the end of the presentations to read those out for our faculty. Tonight's event will also be recorded for viewers who are unable to join us this evening so they can watch it at a later time. And the material will be shared by Simon Fraser University's Faculty of Science. The organizers will also send out an online feedback survey to all of those who attended tonight. We appreciate receiving any comments and suggestions you have.
That's right, Stu. We would love to hear from you, your thoughts and ideas. Please send us your comments and questions on the right-hand side of your screen. Thank you so much for your participation in this event. We would also like to thank Oh Boy Productions for helping us bringing you this event to you while following COVID-19 safety protocols in studio. Tonight, we are happy to have with us 3 faculty members for Simon Fraser University to present this year's Nobel Prize awardees and their research. We are thrilled to be able to stage this event virtually with them.
Now, before we move on and introduce our faculty presenters for this evening, we'd like to hear a few words from the Dean of Science at Simon Fraser University, Dr. Paul Kench. Paul, unfortunately, is unable to join us physically in the studio tonight, but he has pre-recorded some befitting remarks to help us begin our presentations. Let's all hear from Dr. Paul Kench, the Dean of the Faculty of Science over at Simon Fraser University.
Thank you, Stu, and welcome to everyone that is joining us for this special virtual event. SFU Science, in partnership with the science world, is delighted to present our annual Nobel Lectures. This event places the spotlight on 3 of the world's most exceptional science advances, which have been recognised in the latest Nobel Prize awards. At a time when the importance of science is exemplified in our response to the global coronavirus pandemic, this event allows us to reflect on and appreciate the work of scientists and their truly outstanding discoveries. This year's Nobel Awards reflect profound advances in science that have made substantive impacts on our world. The Nobel Prize in Physics was awarded for groundbreaking discoveries about the most exotic phenomena in the universe, the black hole. The Nobel Prize in Chemistry was awarded for the discovery of one of gene technology's sharpest tools, the genetic scissors, which afford researchers the ability to precisely change the DNA of animals, plants, and microorganisms. This technology has had a revolutionary impact on the life sciences, is contributing to new cancer therapies, and brings the dream of curing inherited diseases a giant leap closer to reality. The Nobel Prize for Physiology or Medicine was awarded to scientists who made a pivotal contribution to the fight against a major global health problem. Their work led to the identification of the Hepatitis C virus and made blood tests and highly effective antiviral medicines possible, saving millions of lives. I'm delighted that during this virtual event, you will be hearing from three of SFU's leading researchers who will bring to life these discoveries recognised by the Nobel Prizes, highlighting the enduring impact of this science on our global community. I want to personally thank our presenters for taking the time to participate this evening and for shedding light on these incredible science discoveries. Thank you also to Science World, our community partner, for celebrating the Nobel awardees with us in the community, and to Oboy Productions for assisting us stage this event. Most importantly, thank you, our SFU alumni and donor friends, our science students and SFU faculty, Science World members and community partners for joining us. I trust you enjoy the event.
Inspiring words from Dr. Paul Kench. We always appreciate opportunities such this to engage with the public on science research. And now it is time to introduce our three faculty presenters who are with us this evening at the studio. First off is Dr. Andre Frolov from our Department of Physics at SFU. Following his PhD at the University of Alberta and postdocs at Canadian Institute for Theoretical Astrophysics at the University of Toronto and Kavli Institute for Particle Astrophysics and Cosmology at Stanford University, Dr. Andre Frolov joined SFU in 2006. He is interested in the theory of gravity and early universe and exotic things like extra dimensions. Lately, he became increasingly interested in observational cosmology and since 2013 has been working with the European Space Agency's Planck and now Simon's Observatory and Planned Cosmic Microwave Background S4. Welcome, Andre.
Hello. It's nice to be here. Hello, everybody.
Okay. Thank you, Andre. And our next presenter for this evening is Dr. Timothy Audus from the Department of Molecular Biology and Biochemistry. Dr. Audus joined the Simon Fraser University community in 2016 as a Canada Research Chair, following the completion of his PhD at the University of Guelph, where he was a scientist. Then he went on to become a scientist at the University of Ottawa and the University of Miami. Today, his research team studies protein misfolding events that have been linked to common neurological disorders such as Alzheimer's and Parkinson's disease. Thank you for being here, Dr. Audus.
Thanks for having me. I'm looking forward to having a great discussion tonight with all of you.
Our last faculty presenter for the evening is Dr. Nike Von Hauten from the Faculty of Health Sciences at Simon Fraser University. Nike has a research background in immunology and antiviral vaccine design. She is director of undergraduate programs and a senior lecturer. She is teaching human biology and infectious diseases. Good evening to you, Nike.
Good evening, and thank you very much for having me tonight. I'm really looking forward to the evening.
Well, it's great to have you here, and this already feels like a really engaging evening and discussion session. So again, at this point, we'd like to remind all of our viewers joining us virtually that we have a question and answer field on the right-hand side of your screen. We'd love to receive any of your questions, and comments. And once you send them in to us, we will, of course, get those and then turn them over to our presenter. Karina and myself will have time at the end of all 3 presentations to do that, to read those out loud to the faculty. So thank you for your participation.
Sounds good, Stuart. Let's begin with our first presenter, Dr. Andre Frolov, to present a Nobel Award for physics. Andre's presentation is entitled From Singularity Theorems to Real Black Holes. Andre will talk about the 1965 Penrose singularity theorem, predating the term black hole itself, and what it meant for our understanding of how black holes form, and then fast forward to the present, in which the gravitational pull of the black hole at the center of our galaxy has been exquisitely measured by mapping the motion of the stars around it. Friends, welcome Dr. Andre Frolov.
Thank you. And to give the talk, I'll finally get rid of the mask. And it's a great pleasure to actually talk about something that is sounds like a science fiction, but it made a long story from being a science fiction to something real. And now it's an astronomical object people study, can have real measurements in, and well, make movies about as well. As you know, there is lots of black hole movies around. In fact, some of them actually involve the science people in earnest. So let's have this show going. And the Nobel Prize in Physics 2020 went to 3 people. One half of it went to Sir Roger Penrose, and one quarter went to Reinhard Genzel and Andrea Ghez. So this is shared for the theory part and essentially experimental part. And the quotation for the prize itself is twofold. For the Sir Roger Penrose's part of it, for the discovery that the black hole formation is a robust prediction of the general theory of relativity, and for the 2 observers, for the discovery of the supermassive compact object at the center of our galaxy. So that, of course, is a quote that the Nobel Prize Committee sort of puts out, but as you can see, it is hopefully generic in the part of robust prediction of general theory of relativity. So I want to spend a little bit of time talking about what was all of that about, and what sort of theoretical papers made a big impact into the field. All right. Perhaps I should start with half joke, half real slide. If you have any problem in life, one of the best places to take it, go and have it explained, is to go to XKCD comic website, which has answers to all the science questions. I continuously study there to see if I have any good slides available to me. But anyway, here's a slide which explains the 4 forces in nature. I will not read the joke, it won't be a joke anymore. But anyway, there is 4 forces of nature, which is gravitational force, electromagnetism, then strong nuclear forces, and the weak forces. The latest 2 are domain of people who doing physics on really small scales. The ones which you run into every day is probably gravity. You feel it every time you pull yourself out of bed in the morning. And electromagnetism, although you might not be realizing it, it's chemistry and all the rest of science which you usually do. And well, atomic ones you don't really usually have intuition about, and you go into physics graduate program to study them properly. Anyway, this talk is about gravity and the consequences of the gravity. So 2 main personages in this story is sort of Newton and Einstein. You've seen the pictures of all of them. I'm sure they're quite popular in social networks, although they have a long sort of history behind them. And the understanding of gravity changed a lot between this epoch from Newton to Einstein. I have in the background actually the image of the accretion disk around the black hole ray traced with the proper scientific tools nowadays. So that forms a background to all the story today. It's a story about the black holes really. But to understand how the black holes sort of form and behave, it's actually sort of, you need to understand how the gravity works. And it turns out to be a rather mysterious force of nature. So Newton's law you might have seen in school or in university first year if you go there. The 2 bodies attract itself. The heavier they are, the bigger the attraction. The closer they are, the bigger the attraction. This story got changed a lot in understanding, but increased in richness a lot when Einstein came along. the greatest realization was that the space-time was not something immutable in which the bodies live in, but rather something which is dynamical and has degrees of freedom. So rather than having something move because there is other bodies exerting a force on it, it's the entire space itself that gets bent, and the bodies, including light, react to this change. So in some sense, the main difference between Newtonian and Einstein theory is the nature of space and time itself. So that's a profound impact we have. So I can summarize these things without writing a bunch of equations. Of course, writing a bunch of equations is my favorite pastime, but it's probably not appropriate here. But anyway, so you can sort of envision the effect of the gravity by thinking about space-time being as elastic object. And space itself is warped by matter. So if you have something heavy like Earth or Sun around, it will deform the space around it. If it's rotating, it will drag the space around it as well. And other things which propagate in the space see that the space is actually kind of curved. And the matter moves in a curved space in a straightest possible line, but that straightest possible line might not be straight anymore because space-time itself is curved. So this goes and gives you the usual things like planetary motions, where things go around ellipse, around a sun, which can be measured very precisely. Or extreme cases, for example, like a black hole, and that's the subject of our talk today, is a black hole in the center of our galaxy. So to summarize the theory of gravity, perhaps it's best to remember the quote from John Archibald Wheeler. This is the person who actually came up with the name of the black hole in the first place. And the quote is very simple. Space-time tells matter how to move, and matter tells space-time how to curve. So that's basically a theory of the gravity as we know it in a nutshell. But of course, things are not so simple, and if you go to extreme regions of gravity, you're interested to find new things and to see if the gravity is really like Einstein envisioned, or something more fun hiding behind the corner, just around the corner. We have lots of hints from cosmology saying that the gravity actually might be much harder to understand for the other things. They also don't mesh well with quantum physics. That's a subject of another talk altogether. That's still an open problem. But anyway, let's talk about the black holes. So black hole is the end state of the collapse of a big object like a star that cannot no longer support it because it's burning gases. And when the fuel, for example, burns out, there is nothing to stop the action of the gravity. And if the star is big enough, it will collapse. And the question about what happens after it, when it collapses, has been like, theoretical physics question for ages. And interesting things coming around. When the matter becomes so compact that even light cannot escape from the surface of this object. Remember, if you're sitting on top of the Earth, you can go in a spaceship. Well, nowadays you can even do it as civilian and fly to the orbit. You just have to go fast enough. Fast enough for Earth is about 8 kilometers per second. Fast enough for a compact object like a neutron star would be almost the speed of light. And for the black hole, even the speed of light is not fast enough. So there is a region known as event horizon, which hides the black hole center from you. And if you go in there, even the speed of light is not enough to escape from you. Common day analogy would be if you have something which is limited velocity of swimming, for example, a goldfish, and you put it in your bathtub, but the bathtub goes down the drain faster than the fish can swim. That usually basically means it's a one-way ticket for the fish. I would not advocate doing this at all, but that's sort of how the gravity works for the black hole. So original quotation of this in the Nobel's work is actually talks about Penrose's general theorem about the black holes being generic in the general theory of relativity, which is what Einstein theory is called. So I have this photo of much younger Penrose here on the screen, but it's still way, way after the actual paper was taken. So the photo dates from 80s in Oxford. And the paper itself is 1965. It's almost 50 years old now. Actually, it is more than 50 years old now. So it is actually old. But it is set up a lot of things in modern general relativity and put the context for a lot of discussion about the black holes themselves. The first black hole solutions, for example, the one described by Schwarzschild, came about almost when Einstein wrote his theory. That's basically 1916s. So after that one, people have been grappling around for different black hole-like solutions. And one of the questions was, is a black hole really something which is always appearing when the objects collapse? Or is it some sort of, artifact of the fact that you set up your space-time to be exactly and perfectly spherical? So when you collapse everything, it goes to a point, and the point being like, a complicated object by itself, like, will have to do something to dress itself up. So contribution to Penrose basically was to study this with a toolkit he brought from his understanding of mathematics and prove a simple theorem, which was the first time singularity theorems sort of got named, about the fact that if you actually form a black hole which has a trapped surface, then the only way to go is go and form a singularity inside it. That's what the gravity theory, like Einstein's gravity, will predict. And it is a bit technical, but it illustrates very well by the figure, which I am taking straight from that paper in '65, and it is better drawn than most of the current days figures, so I like it very much. And essentially, it summarizes what happens when the star dies and has its surface shrink under the weight of its own matter collapse, and create a black hole in it. So the diagram has spatial directions running horizontally and time direction running vertically. And the thin line with the light cones and eyes attached to it going left on your screen are the light rays that I escape from the surface of a star as the thing is getting collapsed. So as you form in, the matter falls down, it becomes smaller and smaller in size, and eventually forms a region with a gravity pull so strong that both the outgoing and ingoing light rays actually go towards the center of the space-time. And the theorem that Penrose proved in the basic of a lot of understanding of the black holes right now is that once the light rays start converging, they cannot spread around in the theory of gravity. Gravity is attractive after all, so if the lights are already coming together, then the matter effects would be only to accelerate this convergence. So they will have to intersect, and if they have to intersect inside, they will essentially either cut off the space-time or make sure that you run into something which is known as singularity, which is basically the region where all the matter is concentrated, which basically means we don't know what's going on, something which needs to be done with the physics theory itself. So this has been our sort of picture of the understanding, as a basis of understanding of what the black holes are like. So this concept of trapped surfaces are introduced. Technically, these are the surfaces from which the normal light rays both shrink in area as they go outwards. But essentially, it is the surface where you are inside the surface. Your fate is already decided. You basically have to go inside the black hole, and whatever happens there, I guess, stays in the black hole, unless you have a connection to the outside world, which could be possible. And people studied a lot of exotic things like wormholes, the black holes which open up in the extra universes. If such thing could be possible, that interesting things like time machines could also be possible. There is a huge amount of what sounds like science fiction associated with this picture, but it's not really science fiction anymore. We are accessing the just outside of the black hole with current observations, and we can see the black holes collide, or rather hear them collide, and I'll get to that in a little bit of a second. So that's a serious side of the story. It's taken 50 years of obscure theoretical papers being written by academics to get from the particular solutions like Schwarzschild found to this general understanding of the black hole space-time. And it's taken another 40 years to actually understand how this goes dynamically and how this process of two black holes colliding will actually go about it. But today I want to talk about the big black hole. Well, black holes are usually rather big, being astronomical objects. They might be masses of a few suns, like 10 solar masses. But the one I'm talking about is in the center of our galaxy, the Milky Way. So this one is really, really huge. Well, you usually don't get to fly around in our galaxy, so you might not be too familiar with where we live in it. But essentially, we live on the outskirts of the Milky Way. It is not elliptical, it is actually a spiral galaxy, which basically means if we live on the edge of it and we look on it, we'll see the flat thing on the sky with lots of stars. That's the disk of our galaxy. And you can see it all the time. Well, mind you, maybe not in Vancouver, but if you go outside at night, presumably in the dark environment farther away from the cities, you see this around you if the light pollution is not too bad. So that's a quintessential Canadian picture. It's Milky Way during the summer over the Moraine Lake. I took it this summer. And these things I essentially show you what the night sky looks like if there is no city lights near to you. So the center of our galaxy is actually somewhere at the horizon, this picture. So the only thing you can clearly see on this one is 3 very bright stars on the summer sky, the so-called summer triangle. It's Vega, Deneb, and Altair. And if you actually want to take a look and observation of the center of our galaxy, you will actually have to go somewhere else, not in Canada, because in Canada, it's even at the best observing things, it's almost at the horizon, which basically means you'll have to do better and observe it from some better venue. Some better venue, of course, is high altitude, close to the equator if possible. So there are a few sites which make much better pictures of the center of the galaxy. So here it is, blown up from the big adaptive optic telescope on Keck telescope in the central stellar region. And this is where our hero is, the black hole Sagittarius A star, as it is known. Sagittarius, of course, is the name of the constellation. A star is the object in it. Astronomers like to give cryptic names to the objects they work with. But anyway, to make this story, it's a real story about technology. To figure out the small object in the center of our galaxy, you have to have a huge resolution available to you. So you want to have a big telescope, not only to collect a lot of light, but also to collect a lot of light and make a high resolution on the sky. And if you studied physics, maybe at university, you perhaps heard about diffraction. That's a property of the light being a wave, a property of it to spread around a little bit. So you have to have a big mirror to have a very good resolution for this thing. In fact, even having the biggest mirror you can possibly buy now, for example, like these 10-meter class things in the Keck telescopes, are still not good enough. You have atmosphere moving around you. So if you actually look at the clear, cold night, the stars will twinkle. That's not good if you're an astronomer. Twinkle, twinkle, little star is a very bad thing if you're an astronomer. It basically means the air is moving in front of you and smearing everything. People learn to deal with this, and it's known as adaptive optics. So essentially what you do is you take lots of small exposures with a telescope and try to refocus it by having actuators in the mirror every time for a bright object to stay a point, which basically means the dim ones will now become a better points. And that's a dramatic difference that was sort of enabling for this discovery. It's a discovery of adaptive optics. All right, so the center of our galaxy has been observed for a while, and that is the movie that goes from Andreas Geze's group from UCLA, and that reconstructs the orbits of all the close stars around the center of our galaxy. The star in the center is where the Sagittarius A object is, and all of these cryptic labels of SO, SO, SO is basically central object stars in the center of the galaxy, which can be seen on every plate you take, and people have been doing it since 1995. And this basically means that you can go and take a look how this star moves. And in these 30 years or so, you've actually seen the stars go and make orbits around it and around it. Some of the closest stars, the one which actually has a yellow orbit trailing around it, done more than one full rotation. Its period is about 16 years or so. And it goes very close to the central object. Since it's basically essentially the same story, but much messier one than our planetary system, there is more, there is more elements in here, and it's stars, not planets, which sort of circle around the central objects. You can do the same story we did in our planet and estimate the size of and the mass of the central object. So we know that entire period, entire object is inside the entire orbit of S2. And it goes very, very close to the center. So the close approach it takes, it's only 10 million kilometers. When it goes to the close approach to this object, it's basically 10 times the size of the sun away from the center of this thing. It goes at crazy speed of 7,000 kilometers per second. So from this you can infer that this object, whatever it is, it must weigh about 4 million solar masses. So it's much, much bigger than our sun. But it's only like, confined in the region of about factor of 10 bigger than our sun. In fact, this entire region you take is not much bigger than the solar system we have. So we're pretty sure it's a giant black hole in there. The question, of course, we will ever see it directly or not. And happily for us, we're living in the time where things happening are very fast. And there is entire chance of having this imaged essentially in the next couple of years. We already have the image of the black hole in the center of M87. That's a galaxy next to us. It's a little bit, turned out to be a little bit easier to image using very large baseline radio telescopes like AHT. And it is a fuzzy blob, but it is a fuzzy blob of the things next by the black hole in its very, very close vicinity. So we know these things are there. We can manage their extreme vicinity. In fact, we've seen one in the center of our galaxy to eat up a gas cloud with a little bit of a flare going up. So Event Horizon Telescope is very likely going to image our galactic center as well in the next few years. I don't know what the schedule is. And another thing that's happening even as we speak, it's gravitational wave detectors are operating. So we know the black holes are there. We know how they collide if you have supercomputer big enough and you solve that problem. When they do collide, they produce gravitational radiation, and you can hear this as the minute ripples of the space-time go and propagate all the way across to Earth. You hear those things as essentially direct undistorted voice of the black holes being made. So I'll stop here and basically sort of give you a summary of the 100 years of the black hole's journey. So they started out from science fiction-like stuff, and of course it was going on in academic literature for like 50 years or so as people studied these solutions. People studied about 30 years of galactic center observations now are there in optical and radio. And we have gravitational wave detections a few years back due to LIGO group. And we now have direct black hole images from AHT collaboration, which is another big collaboration trying to image the black holes all around it. So from the things of science fiction, they became real astronomical objects, the things we study and image and collect the data on, and are going to classify and have a lot of information known about gravity in the next few years. So really exciting times, and I'll stop here.
Wow, thanks. Thank you for that fabulous presentation, Andre. Black holes have always been of immense interest to many of us, and they continue to excite our imagination. I know that I was going in so many different directions, listening to you thinking, how do you figure this out? And how do you know that? And where do we go from here? And what do we understand next? And I'm sure many of you who are out there watching right now are forming your own questions about black holes. And so we would appreciate it if you submit some of those questions and comments by typing them into the comments section. And we'll get to those questions and comments later. But for now, it's time to move on to our second presenter.
Thanks, Stuart, and thanks everyone. For very large black holes, we now turn to very small microscopic organisms. Next, we have Dr. Timothy Audus, who will be discussing the Nobel Prize in Chemistry. His talk, called Genetic Cut and Paste, a revolutionary tool for correcting mutations, will describe the development of a new technology, Crispr technology. This new tool allows researchers to better understand basic biological events and potentially develop new treatments for common human diseases. Bringing you back, Dr. Timothy Audus.
Thank you. I appreciate everyone here tonight to learn a little bit about what I think might be one of the most interesting Nobel Prize discoveries of our generation, certainly in the chemistry field. And it's by two outstanding scientists, Dr. Charpentier and Dr. Doudna. And it's for the development of a new tool that will allow researchers and scientists to manipulate the genome of almost any organism on the planet. So this story starts with a rather small bacteria. We just talked about large galactic objects, and now we're moving to microscopic organisms. These are 50,000 times smaller than a loony. And these are called Streptococcus Pyogenes. And I'm going to try and convince you that this bacteria might change the world. You've probably heard of it. Streptococcus is associated with common infections like strep throat, scarlet fever, and gangrene. But today I'm not really telling you about the human perspective of these bacteria. I'm actually going to tell you a little bit about the problems they have of their own. Because like every organism on the planet, they're all fighting off viruses. I think we're all aware of the problems of viruses in today's world. But bacteria have their own viruses, and they look like something out of a science fiction movie. These are called bacteriophages. And bacteriophages actually attach to the surface of these Streptococcus bacteria. And when they do, they inject their DNA into the cell. And that DNA replicates over and over again, produces RNA and proteins. And the whole goal of any of these viruses is to just produce more and more and more of themselves, to a point where the cell just explodes and releases all of the viruses out into the environment to infect other bacteria. Now, this doesn't always happen. Sometimes the bacteria wins, sometimes the bacteria survives, and at those occasions, then you have sort of an immune response that allows the bacteria to protect itself should that virus infect it again. And this is a lot like in how humans, if we're ever exposed to something like chickenpox, we won't get it a second time. And so the bacteria immune system is going to be used and developed when replication is stopped. And so what happens is the little piece of the viral genome is cut out of the DNA. And that little piece of viral genome is transported and it's placed inside what's called the Crispr locus. Now, I'm sure many of you have heard the term Crispr. It's really popular in the media and in the movies. I can be fairly confident in saying that no 30-foot alligators or wolves are going to destroy the city of Chicago using CRISPR technology. It actually refers to clustered, regularly interspaced short palindromic repeats. That's a great Trivial Pursuit question, but it's not really something you need to know. CRISPR itself is actually this region of the bacterial genome where each of those colored boxes represents a different piece of viral DNA from a different infection that's occurred over time. Now, I need to explain a little bit about DNA and RNA for those who aren't familiar, because that's what these loci are made of. Deoxyribonucleic acids, that's DNA, and that's the storage genetic information in the cell. RNA is like the brother or the sister molecule to DNA, because they're both very similar molecules, and they form these long strings. DNA contains 4 different subunits, and those subunits are arranged in different orders: C's, A's, T's, and G's. And the order of those C's, A's, T's, and G's, called nucleotides, are what encodes the different genes in your body. Genes that control your eye color, genes that tell your cells to divide, genes that tell your when you're just a baby to turn into a human and not a monkey or a turtle. And 2 strings of DNA or RNA can come together if they're what we call complementary. Okay, so I've represented it with puzzle pieces. And what I mean by complementary is you can see how the T and the A seem like they would fit nicely together. These 2 strings of DNA are not complementary. You can see they don't fit together like a nice puzzle. But these 2 pieces of DNA, even though their sequences are completely different, starting on the left side, CATG on the top strand and GTAC on the bottom strand, in fact fit together perfectly together. These are complementary sequences of DNA, and I'm going to show you a lot of complementary sequences of DNA because it's critical for how CRISPR functions. I'm going to draw it like this throughout the rest of the presentation, though you can imagine it as the puzzle pieces if you like. And what I really need you to take home here is that complementary binding allows one piece of DNA or RNA to be used to very specifically recognize another sequence of DNA or RNA. So let's go back to our poor bacteria that's being infected yet again with a bacteriophage or virus. Thankfully, it's a virus that it's recognized before. And so now we're going to activate the CRISPR-Cas9 complex. And that's that molecule in the center of the screen. It's made of 3 different biological molecules that I've pointed to with arrows. And if we zoom in, we're going even smaller now. Now we're talking 4 million times smaller than a loonie for this CRISPR-Cas9 complex. And in fact, the ridges and the grooves in that gray structure are almost the placement of atoms within that protein. I'm going to simplify it a little bit, just to make it easier for you to follow. And it's made of this Cas9 protein, which acts as molecular scissors. And that's going to be what cuts the DNA molecule. The second component is tracer RNA, and you can imagine that as almost a scaffolding molecule that holds the Cas9 in place and holds that third component, which is this CRISPR RNA, and that provides that virus-specific guide. the CRISPR RNA is dependent on what virus is infecting it. And in fact, a CRISPR locus can have a number of different CRISPR RNA denoted with each of those yellow arrows expressed from it to target a different virus each time. So imagine like having an antibody against chickenpox or an antibody against COVID. Each of those arrows represents a different sort of antibody or guide for the CRISPR system. Now, once the virus has been infected into the cell, the CRISPR will scan the viral DNA looking for a perfect match. Remember, a perfectly complementary sequence. And when it finds that perfect match that we can see down here, where all of the subunits or nucleotides line up perfectly, then the Cas9 protein cuts the DNA and the DNA is broken. And when viral DNA is cut like this, the virus can't survive. The infection is stopped. And I think you're probably asking yourself, why should we even care how bacteria protect themselves? And wouldn't it be better if the bacteria was destroyed? We'd have less strep throat, we'd have less gangrene. And the answer is, understanding systems like this really allow us to develop tools that could be used to help humans and human health. A great example of that is understanding mold, and the understanding of mold led to antibiotics, which have been used for the last few decades. So, what did Dr. Charpentier and Doudna do? Well, they realized that this CRISPR-Cas9 tool, like antibiotics, could be used to help humans. They made a couple of simplifications because simple is always better. They, instead of making a 3-part system, had a 2-part system where they combined that tracer RNA and that CRISPR RNA into this single guide RNA. And then the next thing they realized was that that CRISPR region, that region that detected the virus, could be completely reprogrammed to encode any DNA sequence you want. So this was the one for the virus we looked at before, but we could change those subunits or nucleotides to any sequence any scientists want, and you could have over 1 trillion different combinations encoded by this CRISPR-Cas9 complex. And what this allows scientists to do is it allows them to cut and find any DNA sequence in any organism in the world. So if we inject the CRISPR-Cas9 complex into a cell, we can cut a very specific piece of DNA in that cell. Now you're asking yourself, isn't cutting DNA a bad thing because it killed off the virus? Well, in cells like our own cells, cutting DNA is just the first step. What happens after cutting is that the cell will actually repair the DNA. And it can repair the DNA, and it's almost like trying to put together a puzzle. And so scientists can help the cell put that DNA back together by providing it a template, providing it that picture to put things back together. And where gene editing comes in is that that picture, while it does have to be very close, the regions highlighted in gray do have to be identical, you can have regions that are dissimilar. And by having a template that's slightly different than the original sequence, you can modify or edit the genome, changing something like a CT base to a TA base. So what can we do with this tool? Well, in the lab, scientists like myself are using CRISPR every day, and scientists all around the world are using it as an important tool to help us understand how the world works, how human disease works. We modify flies or worms or cells grown in a petri dish to help us gain a better understanding of the world we live in. But a flashier use for CRISPR-Cas9 might be to use this tool out in the real world. A couple of applications that I'll talk about are using CRISPR in agriculture. We could use it to genetically modify the food that we eat, the plants or the cattle that we consume, to improve pest resistance or increase nutritional value. But I think most people, when they think about gene editing, they jump to this idea of modifying humans. And I want to start with you and tell you that scientists absolutely don't think genetic modification is going to create people who can shoot laser beams out of their eyes, or climb up the walls, or turn invisible. This isn't what we mean by genetic modification of humans. What scientists mean when we talk about human gene editing is they mean correcting some of the hundreds of different mutations that occur in humans' genes to cause diseases. Diseases like Alzheimer's or Huntington's, cancer or sickle cell anemia. All of these conditions that you may have experienced in your life or someone you know has experienced are caused by the mutation of genes. I'm going to give you an example in the next couple of slides how CRISPR could be used as a gene editing technology to cure sickle cell anemia. This is actually being used, this not exact approach, but this is being used in a very limited set of people and patients in the world. So, sickle cell anemia is caused by a mutation in the DNA that leads to the creation of a protein that doesn't work properly. It's actually a single nucleotide or subunit substitution moving from an A that's in healthy individuals to a T that's in sickle cell anemia patients. And that doesn't seem like a big deal because there are 3 billion subunits, 3 billion nucleotides in the human genome. So why would change one nucleotide really matter? And I want you to think about it in terms of what would happen if you change one letter in a word. It can completely change the meaning of that word by just changing one letter. And just like English, changing a nucleotide in the genome of an organism like a human can have a remarkable effect. So in this case, this mutation causes normal hemoglobin, which is the protein that transports oxygen throughout your body, to have a slightly different structure that I've highlighted with these little red nubs here. Now, normally hemoglobin is floating freely around in the cell. You recognize red blood cells here, which are floating around in the tissue, in the blood vessels of your body. And this sickle cell anemia mutant causes those hemoglobin molecules to clump together. Right? 2 can clump together, then 3, then 4, then 5 can clump together, and they create these long strings of hemoglobin. And those strings stretch out those red blood cells, taking it from that flat structure to this sickle-shaped structure. And when they become sickle-shaped, they don't pass through the blood vessels as easily. The body kills them off, and you become anemic. You don't have enough oxygen brought to your tissue. You become fatigued. You become weak. So how can CRISPR help this? Well, normally, before CRISPR, the cure for sickle cell anemia, which isn't used very often, involves a bone marrow transplant from a compatible donor. And that's problematic for a couple of reasons. Compatible donors are really hard to find. You've probably all heard about the trouble people have finding a bone marrow donor for any particular patient. But let's say we can find a bone marrow donor. The bone marrow is extracted from the donor. The sickle cell anemia patient would have their own bone marrow irradiated. It would be killed off. And then it would be replaced by the bone marrow of that healthy individual. Now, there are problems here above and beyond just finding a compatible donor. You also have the issue that you could reject the transplant, and you have the issue that you can be on drugs, immunosuppressive drugs for your whole life. How CRISPR can help is that CRISPR allows doctors and scientists to genetically modify the patient's own bone marrow. So in a way, the patient becomes the donor. And there's no need to find a matching donor to donate the bone marrow. In a lot of ways, we'll extract the bone marrow, and that's common to both procedures. But after that, the CRISPR-Cas9 system is injected into those bone marrow cells along with a template DNA. That template DNA can change that T in the sickle cell anemia gene into that normally healthy A, making that normal healthy hemoglobin protein. And then scientists will select which cells have the mutation in them, and they'll only allow those healthy cells, those corrected cells, to grow up and generate more bone marrow cells. Now, after we've got a lot of bone marrow cells, then the patient, who is also the donor, is irradiated just as before, and those cells are injected back into the patient. And those are now healthy bone marrow cells that won't produce mutant hemoglobin, that won't produce that sickle cell condition. And that gets around this idea of finding a donor, and it also gets around the idea of a rejection because it's your own tissue that's been fixed and put back into your body. So it's a really powerful tool to be used for medical treatment of human disease. But it's not ready yet, right? And there's still a lot of reasons why we can't use CRISPR-Cas9 technology to treat a lot of diseases. And we can talk about those more if you want at the end by just adding a comment to the comment section. And that gives us a lot of time to deal with really maybe a larger issue here, and that's the ethical implications of gene editing. And scientists aren't always just thinking about what can we do, we're thinking about what should we do as well. Now, the first question that comes to mind when you're thinking about gene editing technology is, in what organisms is it acceptable to manipulate the genome? There's a whole spectrum of ideas on what's acceptable to do. I would say gene editing has been happening for over 2,000 years. It's not been in as direct a way as we can with the CRISPR-Cas9 technology, but you may not be familiar with the wild mustard plant. But you're familiar with its offspring, because over the last 2,000 years, the wild mustard plant has been selectively bred for larger flowers or larger leaves, and it is actually the plant that you consume when you're eating broccoli, cauliflower, cabbage, or kale. That's been done over a 2,000-year span just by breeding plants to different plants. CRISPR technology allows us to be very selective in how we evolve our plants. And we can change things that allow something like broccoli, cabbage, kale, or cauliflower to grow in drier areas of the world. And the shrinking farmland that we have and the growing population that we have in the world requires us to produce more food, more nutritionally packed food. And this technology could allow us to do things like that. Now, if you're thinking about genetic modifications and gene editing, you're inevitably going to move towards, should we use the CRISPR-Cas9 tool to modify humans? And I think there's, I'm going to pose two questions on that question, because I don't have all the answers on this. The scientific community doesn't have all the answers. This is an open debate. One of the questions is, what characteristics should doctors be allowed to change? So any genetic modification could theoretically be changed by the CRISPR-Cas9 technology. Any gene alteration that you want to make, you could potentially make within reason. Most would agree, probably most watching us right now, would agree that correcting things like cystic fibrosis, muscular dystrophy, Huntington's disease, and diabetes would be all valuable uses of this technology. But opening this up is a slippery slope. At some point, you're moving from health changes to aesthetic changes. Down that slope, there are communities of people that have dwarfism, deafness, and blindness. They don't feel those are diseases. They feel that they're a part of a community. And so genetically modifying those may not be something that we should be considering doing in the future. And then if we move farther down the spectrum, things like eye color, strength, weight, baldness, all of these can be manipulated using CRISPR-Cas9 technology. And I would argue that it would be much more of a stretch to say we should use the technology to change someone's eye color than we should use the technology to change and correct cystic fibrosis. The next question is, who should be treated with this gene editing technology? And that comes down to adults or embryos. Embryos are what turn into babies, that turn into new children. Most scientists, at least right now, are in fair agreement that gene editing technology should only be done on adult cells. CRISPR should only be given to people who can provide informed consent, know what they're asking for, know what they're doing. And it should never be used on cells that could be passed into the future generations. So that technology and that modification, that dies with the patient that it was used upon, not their future generations. Maybe in future years they'll view that the modification of embryos, that we'll have a change of opinion on that, but that involves a discussion with a broader community of stakeholders throughout the community, of doctors, scientists, politicians, and the general public as well. So I'm just going to finish my talk by saying Dr. Charpentier and Dr. Dudena's invention is really changing the way that we do biological sciences. Researchers are using the CRISPR-Cas9 tool, but they're also unlocking the potential of this CRISPR-Cas9 tool to modify the genomes of all sorts of organisms. That's perhaps the simpler question. The bigger question is, what should we do with this tool? And that question really depends on our values as a society, and that's important why we have talks like this tonight, and we have discussions like hopefully we'll have this evening. And I'd be open to any questions that you might have after the talks are done. So thank you all for your time, and I'll send it back to our hosts.
Wow, I can hear keyboards clicking away right now. I'm sure there's going to be a number of questions that are going to come forward. Thank you for that very, very interesting presentation. And of course, you know, we know that there's going to be some amazing breakthroughs that are going to come with this new technology over time. And once again, I go back to you, our viewers, and ask you to put together your questions, send them in so that we can ask them of our presenters tonight from the faculty. Corina?
We are now down to the final presentation of the evening. Yes, moving on from mutations and new treatments to medicine and physiology. Our next speaker is Dr. Nike Von Hauten from Health Sciences. Nike's presentation is entitled Needle in a Haystack, Molecular Luck and the Discovery of Hepatitis C Virus. For decades, receiving a blood transfusion came with the risk of acquiring a deadly, unidentified infection that caused cirrhosis or liver cancer. A collective 30 years of research led to the identification of hepatitis C virus as the infectious agent. The discovery process required exceptional persistence and the development of techniques that could identify tiny viral components in a relatively huge pool of blood, essentially finding a needle in a molecular haystack. Allow me to turn it over now to Dr. Nike Von Hauten.
Hello, everyone. It's great to be here tonight. I want you to take a moment and think about the smallest thing that you can think of. And I bet it's not going to be as small as hepatitis C virus is. What we're thinking about is something that is 1/1000th of a meter. Scientists might use the term nanometers, which might not be a unit of measurement that you use in every day. And now think of something like that swimming in a sea of blood in very small quantities and trying to understand what it is. How do you get at that? How do you find it? How do you identify it as it's wreaking havoc and causing damage on people's bodies? One of the reasons that this topic is really inspiring to me is because I know a woman, a friend of mine, Anna, who suffered a needle stick injury in the early 2000s while working a custodial job in a lab. And hepatitis C virus is an interesting virus from a transmission perspective. It can remain infectious in dried blood for up to 4 days. And hepatitis C it causes acute disease in a number of people. 55 to 86% of people who get hepatitis C will develop chronic illness and eventually can lead to cirrhosis of the liver as well as hepatocellular carcinoma. So it's a pretty serious illness. Anna was eventually cured as a result of the work that we're going to talk about today. But she's still suffering from long-term damage. And I just want to give her a little shout out. Her birthday's in 4 days. So what we're going to do is we're going to go on a journey through time. And the context for our story is about the blood supply and about safe blood supply for transfusions. And we have to jump back to the 1960s and we have to think about what blood supply was like at that time. And at that time, getting a blood transfusion was actually a very high-risk activity. So people who had to undergo multiple transfusions for various illnesses or people who had to undergo surgery had a 30% risk of developing different kinds of hepatitis following these transfusions. Now, a lot of work was being done at that time to uncover what those what those different forms of hepatitis were. In 2020, the Nobel Prize in Physiology or Medicine went to 3 different researchers. So these are Harvey J Alter, Michael Houghton, and Charles M. Rice for the discovery of hepatitis C virus. Now, this was actually the product of 3 decades of research. And I'm going to take you through each of those decades and each of their research contributions. And I'm going to show you the original data that they published of those groundbreaking discoveries. And I want to point out to you that at the top left of this slide, you'll see a schematic of what the hepatitis C virus, as we understand it now, looks like. And what you'll notice is that there is a nucleic acid genome. In this case, it's single-stranded RNA. Yes, viruses can have RNA genomes that is surrounded by a capsid made out of protein, and that is further surrounded by an envelope made out of lipids or fats. Now, these discoveries did not happen on their own. There was many people, many animals that contributed to these discoveries, and it's been acknowledged by the award winners that it's unfortunate that those people cannot also be recognized by the caliber of this award. We're going to start our journey in the 1970s with Harvey J Alter. And ultimately what he did was he was working with on developing safer blood supply, safer blood transfusions. He had done a bunch of work in the 1960s with Dr. Bloomberg, who eventually won the Nobel Prize for the discoveries of hepatitis B virus, which was a major contaminant within the blood supply. And so he was aware of hepatitises that were in the blood supply. So some of the things that he was doing at the time was he was following patients that had received blood transfusions, looking at whether those transfusions had either hepatitis A virus or hepatitis B virus, and then noticing that there was a proportion of people who were still getting a version of hepatitis even when that blood had been, even when those other viruses had been removed from the blood sources. So there was all these examples of people who were getting some form of hepatitis, and that hepatitis was not related to hepatitis A or hepatitis B. And he also ruled out some other viruses like cytomegalovirus and Epstein-Barr virus, which can be associated with a hepatitis. And so he made the conclusion that there was something potentially infectious within the blood supply that was causing hepatitis, but he didn't know what it was. And so he called it non-A, non-B hepatitis. NANBH. And so in order to show that that was a potentially infectious agent, he did an experiment. And this was a critical experiment, and I'm showing you the data for that experiment on the left-hand side of this figure. So you can see these graphs, and so what he did was he took plasma or serum from individuals who had non-A, non-B hepatitis, and he injected that directly into chimpanzees. And then he watched, then he took blood samples from those chimpanzees over time. So what you're seeing on the horizontal axis is time in weeks, because this virus has an incubation period. And then along the vertical axis, you're seeing the levels of liver enzymes, which can be an indication of a hepatitis happening. And you can see that for many of these chimpanzees, this is going up after about 10 weeks. So this is very consistent with the form of non-A, non-B hepatitis that was happening in these blood transfusion patients. So that was a key discovery. We now know that there is a non-A, non-B hepatitis. And because of Alter's work with other viruses, he had a very strong hypothesis that this chronic hepatitis was being caused by a virus. But by 1988, many people were on the search for this particular virus. They still had not found the specific agent. And you could see that he was quite frustrated at this time, because he wrote this poem. And I'm going to read this poem to you. I think that I shall never see this virus called non-A, non-B, a virus I cannot deliver, and yet I know it's in the liver, a virus that we often blame, but which exists alone by name. No antigen or DNA, no little test to mark its way, a virus which in our confusion has forced us into mass collusion, to make exist just by exclusion. But is it real or an illusion? O great liver in the sky, show us where and tell us why. Send us thoughts that will inspire us. if we don't publish, oh, let us see this elusive virus. If we don't publish soon, they'll fire us. So the eternal scientific dilemma. So now we're in the 1980s. And along comes Michael Houghton and Ki-Lim Chu and George Koo at the University of Chiron. Chiron is a pharmaceutical company that has some degree of academic culture and thus is sometimes coined in this way. And I'm showing pictures of all three of them at the bottom because Michael Houghton has been strongly advocating for the recognition of his colleagues in this award to the point where he has actually turned down some awards because of the importance of their contribution. So they were searching for many years to try to find the actual causative agent of hepatitis C. They were using a number of different approaches and techniques. They were working multiple hours, like really long hours, not taking weekends, because as every day that they didn't find this, more and more people were getting ill. So there was a strong pressing need for them to find this. Now what they ended up doing was taking some plasma from chimpanzees and screening that with some serum antibodies from an infected individual and finding the HCV genome. That's what the figure says. But we're going to dive into that a little bit more deeply because it comes back to that whole needle in a haystack concept. So what they actually were dealing with was this idea that there was really low levels of virus that were present in the blood. And they were looking for ways to concentrate that potential virus. One of the ways that they did that was by spinning the blood or spinning the liquid portion of the blood down, not with the cellular components, hypothesizing that the putative virus would be in that liquid portion, in an ultracentrifuge. Now, ultracentrifuges run at tens of thousands of revolutions per minute, and to get a virus to go to the bottom, you have to spin that down for many, many hours. So they did that, and they ended up with these little pellets. So that's the first part of concentrating the virus. And then they made what they call a DNA library. So they undertook a number of steps and took all of the genetic material in that pellet and made a whole library out of all that genetic material. So basically you're looking at all of these random fragments of nucleic acid that are encoding different bits of nucleic acid that are found in that pellet. They had a million different clones. The system that they were using was a bacteriophage expression system, kind of like what we saw in the previous talk. And so that linked the nucleic acid sequence to the actual protein. And then the serum that they were using from infected people to screen those were then able to detect the protein, and then they could investigate the nucleic acid sequence associated with that. Now, out of those 1 in a million, they found one clone. They found one reactive clone. And that clone turned out to have a portion of the hepatitis C genome, and they were then able to expand on that and build it out more. And so what you're seeing here is one of the key pieces of data. It's called an Immunoblot, and there is a piece of protein that is being expressed from the hepatitis C genome, which they have now officially named. And in that middle section where you can see just underneath non-A, non-B, NANBH, you'll see that little dark spot that's forming. That's the indication of a chimpanzee who's been infected with non-A, non-B hepatitis reacting with the protein that came from that one clone. And the other ones, hepatitis B virus and hepatitis A virus, they are not reacting. So that is showing this positive result. Super exciting. Now, the identification of the virus and the viral genome in this way was a critical insight. But then the translation of that into a practical test was the other part of Houghton's work. And so they used that same protein that I showed on the previous slide to start screening all of these other people that had, that had been acknowledged to have non-A, non-B hepatitis, and they were all coming out positive. So they were now able to link the disease that they had with this particular genome sequence. Very critical discovery. So that was the 80s. And now we're getting into the 90s with Charles M. Rice. And you might be thinking, well, what is there possibly left to do? Well, by 1997, a lot of the work done by Houghton had led to a lot of characterization of the hepatitis C virus genome, but there was still a lot to know about the structure of the virus and the functional genome of the virus. And there was a lot of issues in terms of working with the virus itself in the lab. So, the virus had poor replication in cell culture. There's a restricted host range for how you could work with the virus. And so having a functional genome clone was important. And also to this point, the actual causation of having that genome cause the disease had not been done yet. So it was associated with the disease fairly strongly at this point, but showing that the genome actually caused a hepatitis on its own had not been done. So this is where Charles Rice came in. And he was trying to identify these functional genome sequences of the hepatitis C virus. And this was a challenging process because when you have an RNA virus like hepatitis C virus, it doesn't have what we call good proofreading capacity. So the virus makes a lot of errors when it makes new copies of itself. So you have all of these different sequences within a pool of that particular virus. So he extracted extracted thousands of copies of genomes from a particular individual. And then he narrowed that down to 233. And then he narrowed that down to another 34. And then he took those 34 copies of genome and he injected them into a chimpanzee. Or maybe a few chimpanzees, whatever. And guess what? It didn't work. So he had to go back to the drawing board and look at all of these sequences again and analyze them again and redesign them. And that's what you're seeing on the left-hand side of this figure, is the modified versions of these hepatitis C genome sequences. And then he took all of those that he'd then modified and injected those into 2 chimpanzees. And what you're seeing in panels A and B on the right-hand side of this figure are chimpanzees that have been injected with these full-length genomes that are now starting to show elevated liver enzymes. They're showing viremia. They're starting to show serological responses, like producing antibodies against the virus, that are very typical with hepatitis C. So we have now confirmed that the virus is now, that genome of that virus is now associated with hepatitis. So collectively, we've just gone through 3 decades and 3 to 4 major contributions. We've identified an infectious non-A, non-B hepatitis by exclusion in people who receive blood transfusions. We've seen the identification of the virus genome. We've seen the creation of a diagnostic test, and we've confirmed that the hepatitis C virus genome causes non-A, non-B hepatitis. And collectively, these discoveries have led to really important diagnostics, as well as targeted new drugs. But we've really focused on blood transfusions here because that is what has driven this work. Hepatitis is also passed on by other means, and it is a critical global disease that infects 71 million people and causes 400,000 deaths per year. And so even though there are these treatments, if you have access to health care and money, it's not available to everyone, and in order to have that, that requires committed effort. And so there's a strong call to use the momentum from this prize to enhance that initiative. Now with that, I just wanted to say thank you to a few people, the students of my Health Sciences 482 senior seminar course, who we've actually been going through this as a case in our CAT class, my friend Anna, and the SFU Faculty of Science.
Wow, Dr. Van Houten, thank you very much. That was incredibly insightful. And certainly a lot of our viewers are curious to learn more about these viral discoveries, especially during a pandemic crisis such as the one that we're in today. It is also exciting to see how the questions are rolling in. We've got quite a number of questions coming in from our virtual audience. But for now, we're going to hold off for just a little bit longer before we get to read all of your comments, but do keep them coming. And thank you to all of our faculty presenters tonight from Simon Fraser University for your insightful presentations. Your enthusiasm and passion for science is clearly evident and gets me excited about science. So before we move on to the questions and comments sent in, I know because many of you in the audience are out there are aspiring scientists, you may yourself be saying, how do I get into a field similar to the ones that are expressed by tonight's experts, or something completely different? Well, this is my opportunity to tell you that if you want to grow your understanding of our world through scientific pursuits, you'll find an incredible community of professors, researchers, and students in Simon Fraser University's Faculty of Science. Their programs are designed to inspire students to make real-world changes through their work, from doing a co-op work term, to partaking in research projects, to making a difference in their communities. Karina?
That's correct, Stuart. If you would ever like to learn more about studying science at Simon Fraser University, we at the Faculty of Science encourage you to view our website at sfu.ca/science or connect with our colleague Claire Wilson via email at science_recruiter@sfu.ca. Claire can answer any question you have. We hope tonight's presentations inspire you to be scientists too someday. Now it's time to hear from our audience. We appreciate you staying on for this segment. Let's take a look at those questions and comments you have sent in.
There's quite a few of them. Karina, you want to ask the first one?
Yes. So the first question goes to Dr. Audus. Could you please expand on the technological, not ethical reasons why Crispr is not yet ready to be applied to fixing human disease problems?
Yeah, thanks, Randall, for your question. I alluded to this in my talk, and actually there's a whole slew of reasons why we're not ready to use CRISPR to fix human disease. I'm going to kind of break it into maybe three categories. The first is that the CRISPR technology that we have right now, the tool that we have, still makes a lot of mistakes. So CRISPR, while it's supposed to cut a very specific sequence of the DNA in the genome, has a tendency sometimes to cut where it's not supposed to cut. And when you start cutting DNA in unfortunate places, that can lead to terrible things like cancer, or it could cause more problems than it's fixing. So that's a big limitation. Some of the new technologies are being used, are being developed to make it more faithful in its targeting of a specific region. Another reason is that right now we don't necessarily know enough about all the diseases that we want to try and treat. So the technology is advancing so fast that we need the basic research that labs all around the world are doing to understand why why a disease is acting the way it's acting within the body. So we need to catch up a little bit on that basic research to where we are on the health side of research. And then the third one I'm going to mention is it's really hard to get that CRISPR into the cell. So if you want to treat a disease like progeria, for example, I'm not sure if anyone knows the name progeria, but you've probably seen pictures of people suffering from progeria. That's premature aging, where you see those poor children who look like they're 70, 80 years old. And so if you want to treat progeria, it's actually a simple substitution, very similar to what sickle cell anemia was, where a single subunit or a couple subunits are changed. And the difference is, with sickle cell anemia, you can take the tissue out, modify it, check to make sure you got it right, and then put it back in. With something like progeria, you have to treat all the tissue within the body. We call that in situ, within that individual. And say if we just manage to treat the hand and not the entire body, well, they might have a young-looking hand, but the rest of their body will still look like it's prematurely aging. So to get the CRISPR technology all throughout the individual is really challenging, and to make sure that it's working properly when we do it in that setting is really hard. So I think a lot of the blood-based diseases will be potentially treated first, where we can take bone marrow out, things like thalassemia and things like sickle cell anemia, where we can take the bone marrow out, treat it, and put it back in, is likely the way CRISPR is going to go at first. And then we'll eventually have to devise and develop technology to deliver CRISPR into all the cells of the organism that we're trying to target, whether that's humans or whether that's livestock. And get it in there and make sure it doesn't make any mistakes. They're working on CRISPR, they're modifying it so that instead of just cutting the DNA and walking away, it actually stays attached to the DNA and modifies the nucleotides, modifies those subunits while it's bound to the DNA. But it's still a long time away, and any medical technology does take years and years to do. The development of any drug that you've probably gotten over the counter or been prescribed by your doctor can take 10 years, 20 years to be developed. And so this technology isn't something that widespread is going to be used in the next 1, 2, 5 years. This is going to be a long game, and it's an important first step, but we all do have to remember it is a first step to this process.
Yeah, it's been more than 20 years since we mapped the human genome, and we're still just starting to work it through the system to be able to say, okay, how can we benefit from this knowledge? So let's switch gears, Andre, to you. There's a question that's come in, which is, could there be an increase in the nuclear forces where the stars begin to implode because temperature will increase with the stars again?
Yes, absolutely. Great question. And there is a lot of stories that I skipped about this one. But in principle, if you start compacting the star, it will heat up again, so it can start burning again the heavier elements. And eventually, even after you run out of the fuel for it to burn into thermonuclear fusion, there still may either things like nuclear forces holding it together. One of these, or rather two astronomical objects that come about like that are white dwarfs and neutron stars. So these are the ones which are stopped from collapsing by quantum pressure of the matter being really, really dense. So a neutron star is much denser than essentially nucleus densities in the normal materials. It's literally like, one big nucleus squished together by the force of gravity. So there can be very exotic forces there. We don't know much about what the forces will do at this, subnuclear densities even. So one of the ways of people trying to figure out what's going on with those stars is find them, measure their size. That you can do if you can figure out how bright it is. And measure the gravitational pull. This allows you to infer the equation of state for the nuclear matter inside. And people are very much open to the possibility that there might be very exotic objects in there. Maybe not a neutron star, which is made mostly of neutrons, but maybe something like a quark star, or something entirely else, different from Einstein's theory. So one of the things that people are excited right now is trying to figure out if the black holes we get in real life are the ones which are Einstein black holes. So maybe something interesting nature has for us as a surprise. And if it's coming, it's going to be big. So, and it's coming in the next 2 years.
Another question now for Dr. Van Hauten. How do scientists overcome the problem of hepatitis C transmission during window period from blood donation?
Thank you for that question. So first of all, let's break down the question a little bit in terms of what it means. Usually when we're talking about window periods, we're talking about a period of time in which infection has occurred and the process of seroconversion happens, meaning there are detectable antibodies that emerge to that infectious agent within the blood that can then be detected. So the question is, how can, and if you recall with hepatitis C, we saw even like within the experimental data that I showed you, there's multiple weeks where an individual could be positive for hepatitis C, but not show antibodies yet. And so then the question becomes, how do we create safe blood supplies? And the answer is, I am not actually that sure how the tests work right now. So this is assuming that there's an antibody-based test that is detecting for hepatitis C in screening of blood. I'm not actually sure if blood screening for hepatitis C works that way. It could be that there are other more sensitive tests that have been developed in that time. And so sometimes when I come across a question like this, like when I'm working with my students, they ask me questions all the time that I don't necessarily know the answers to. And so then the process becomes, well, how can we find that answer out? And so there's a few ways you could look this up. So if I were in front of a computer right now, I might start looking at like Canadian Blood Services and looking at some of their more in-depth information about what their screening processes are. If they don't have the information, I might have to go to something a little bit more scholarly. You might have access to Google Scholar, or I might go to PubMed, which is a really important resource, and say, what are the ways that we can screen blood for hepatitis early on? So for hepatitis C specifically. So those are some of the things that I would do to look up the answer for that question specifically.
Andre, back to you. Kepler's laws, which were established a number of years ago, about 300 and some odd years ago, would they be valid in determining the mass of Sagittarius A using orbit of S2?
Thanks, another great question. I should say that Kepler got in a lot of trouble for those laws, not as much as Galilei. But anyway, so the answer is mostly yes. So in physics, the laws don't just stop working right away. They have domains of applicability, right? If you start going to more and more interesting places, the laws will start break down, but they will break down a little bit first. So Kepler's law, in particularly in our solar systems, are very well tested. So planets do go around on elliptical orbits with a little bit of change. For example, if you look on Mercury for too long, you'll notice its orbit axis is actually slowly drifting with time. So rather than going around on an ellipse like that, it kind of goes around on an ellipse, which sort of gyrates. So this is happening for all the objects because of the relativistic corrections. And exactly by how much this perihelion of the orbit will advance or retrograde, it depends on what the theory of gravity is. So people are extremely interested in measuring out these small deviations from the elliptic orbits, and I'm sure it's going to be done in the galactic center, if not already to the great precision it is done now. And there are even more compact systems. For example, there is actually 2 neutron star systems which was been found accidentally about 10 years ago, which have the signal from the 2 of these neutron stars, and they have about 1.5 hour period, so moving really fast. As opposed to tens of years. And people are measuring the deviations from the Kepler law there. And that's one of the goals in science. Go down, drill down in precise measurements, see if the orbital motion of things, like the stellar motion of the things is like Einstein's theory or not. And in particular, this also goes for black hole collisions. Are the black holes really like Einstein black holes? Do they collide like Einstein black holes would? Or are we looking at something else, some new physics in its entirety? Well, if you're getting something new, you'll hear it. So,
yeah. Wow.
Another question, I think maybe directed to Dr. Tim Audus. In future, much of the bacteria would surpass antibiotics. Could this method, I guess Crispr, be used to eliminate the need of medicine, say in 20 to 30 years down the line?
That's a great question. I would argue that bacteria have surpassed most antibiotics already. So that's not the future that we're discussing. That's the present. Many bacteria, and we've heard of them, are antibiotic resistant to a whole panel of antibiotics. So it's something we do need to really think about a lot in the future. That's not exactly my area of expertise, but I have heard discussion about ideas like using bacteriophage that are encoding for CRISPR RNA to inject into the bacteria an actual CRISPR RNA that the own bacteria's immune system will use against itself and cut its own genome. So there are possibilities that this CRISPR technology could be used against the bacteria. Modifying the bacteria to not produce the CRISPR, things like the Streptococcus, would be more challenging. And I'm not 100% sure that CRISPR will be the main driving force as a replacement of antibiotics in the near future. If the question is kind of more broadly stated, will all medicines be replaced by CRISPR down the road, not just antibiotics, then I would say that that's unlikely. I think there are a lot of reasons that we would want to treat with antibiotics as opposed to manipulating the genome of a person as kind of our starting point. So I think it's a really interesting question and a really interesting area of research that scientists are looking into. But I don't think we're going to be giving up our aspirin anytime soon and replacing it with a pill of CRISPR. In fact, I don't see CRISPR being delivered in pill form really anytime in the near future as well. This isn't going to be just something where you take a pill and you're cured and you've got a CRISPR pill. This is really the generation of what doctors are calling personalized medicine. And personalized medicine isn't sort of big pharma medicine. It's actually driven in a lot of ways by scientists and research communities where each treatment has to be specific to a certain person. That's an interesting area right now that's growing, and I certainly see a role for pharmaceutical companies in CRISPR technology, particularly in the delivery of the CRISPR technology. But I unfortunately don't think we're going to be giving up traditional medicines anytime soon.
Well, you demonstrate just how complex anything is to do with, of course, our bodies, our health, our well-being. And, you know, Dr. Van Hauten, a question to you. You titled your presentation, you know, Finding a Needle in a Haystack. Why did you feel that it was important to frame it that way? Is it because of the complexity of any of this kind of research and how diligent everybody has to be in their research to try and find answers that can be hiding from you?
Thank you for that question, Stuart. So when I was thinking of needle in a haystack, I was really thinking about the research processes and thinking about the vastness of the number of molecules that were happening in some of these experiments in order to extract a single one. And I think there's a couple of examples, like the work that was done by Houghton in terms of building that library, that DNA library that had 1 million clones. Extracting one correct clone is to have the conditions be there and have everything come into play to get that one single clone is really, I can't imagine from a scientific perspective, like having that moment of, oh, we got something, and you can't see it, but you know, the scientists around me are going, that is the moment. That is the reason that you're doing this. So that is one example. And then also the work by Rice, like he was combing through thousands of different copies of that RNA genome and narrowing it down systematically more and more to getting the right version, and then ending up with 10, some of which presumably were infectious. So, you know, the number of, just the sheer volume, the numbers of molecules that were being dealt with, and the luck and to be able to extract that single molecule from this pile is really why I chose that title.
You know, I'm reminded, I was told once that Isaac Asimov once said, great discoveries don't come in some fantastic eureka moment. They unfold over time, and you look at something and you go, well, that's kind of odd, and it then pushes you back down another line of discovery. Do you find that?
I think that could definitely be the case. I think with research, it is something that definitely builds over time, but then it's going to be punctuated with these kind of pivotal moments. So that pivotal moment in Houghton's work of finding that single clone would not have been possible without the years and years of like systematically trying all of these other different approaches, which were probably a very long, tedious slog, if you will, to try to find that. And so I think you know, there are these like these clear moments of we have it, but then that build up over time is, you couldn't do it without those build up times.
Yeah.
I think it brings a good point to the contributions of the people who worked with the Nobel Prize winners as well, and that these you use the term eureka moment. And certainly for the Nobel Prize in Chemistry, there's a whole community that was working around Drs. Doudna and Dr. Charpentier that were working on these projects as well. There wasn't a eureka moment for that. It was the slow build of scientific information, this development of basic science and basic understanding that really is what allows these great accomplishments to take place. You know, whether it's advances in DNA sequencing technology that could be used for identifying a new virus, or whether it's studying a simple bacteria that no one seemed to care about for years and years. Basic research is what drives the idea of this translational medicine, this medicine that can help people like our viewers, that can be driven into cures, that can be driven into drug developments. And so there's not a huge number of eureka moments, but I will agree, there have been moments even in my own research where I'll see a Western like you were, a blot like you were describing, and just throw my hands up in the air, or have my grad students run into my office just to show me something really exciting that they just did. And these small moments are things that really drive scientists like myself, and drive scientists like, I don't want to put myself in the same category as these great Nobel Prize laureates, but I'm sure it drives them as well.
Yeah, I've had those moments too in the lab, and it's been like, it keeps you going. It keeps you going.
Let's go back to another question about black holes, in the hope that maybe we can possibly make an artificial black hole. What do you think, Andre? Is it possible to make something very dense, matter very intense, and call it an artificial black hole?
Well, at present level of technology, we don't have much spaceflight capability. The trouble, of course, with making such a thing would be, it would be great, but you would like to store it somewhere. And being, you know, dominated by gravity is going to be pretty hard to store. Anyway, to do this thing artificially, you'll have to squish the amount of matter into the volumes which are really ridiculous. If you talk about the entire sun, if you take the entire mass of the sun, it has to be something like 3-kilometer radius region. You have to squish it in. That's substantially smaller than the current, like, you know, 1 million kilometer radius of the sun. So your better choice might be to actually flying around and finding this thing already made for you. It's the strong force you need to squish things together, and gravity on large scales provides this great force, which is probably, like, you know, is going to be the major cause of production of the black holes in the universe. People sort of worried a lot about this in the context of extra dimensions, because if you have extra dimensions like fifth dimension or something like that, the gravitational force might be different in the dimensions, and then it opens up a possibility of really tiny black holes. And people worried about making them an LHC, if you remember that big worry of well forgotten 20 years ago or something like that. That didn't happen, so we haven't seen those things appear. So there is no evidence of extra dimensions so far, and the black holes seem to be produced mostly through astronomical processes. There is still talk about primordial black holes produced during the birth of the universe, but nobody never seen them. And people are on the lookout, maybe they're out there. So that's an interesting mechanism for them to be made, that's for sure.
You know, you use two interesting words there, yet and so far. And isn't that so much of what science is about? We don't know this yet, but we continue to strive to understand what the application will be, or whether or not our hypothesis is accurate, and even if it isn't, it may push us into another direction. You know, I want to come back to talking about CRISPR right at the moment for one of the questions that come up, because people are asking those what-if questions as well. You know, could CRISPR be used to interfere with viruses like hepatitis or HIV, and would it help in the production of, you know, vaccines? You know, what could the role be for CRISPR?
So I think CRISPR has a role with a particular type of viruses, in particular retroviruses. Retroviruses are a class of virus that basically, once they've infected a cell, they insert their own genome into the organism. That's why things like HIV, which leads to AIDS, is such a hard thing to treat, because it goes into what we call a dormant state. And so CRISPR could potentially be used as a way of targeting that viral DNA once it's in the genome of your cells, and damaging it. Because I demonstrated in my talk a way of modifying it by putting a template DNA. CRISPR can be used another way, where if you just simply put the CRISPR molecule with Cas9 in there with a target sequence to something like the HIV genome, it'll cut the DNA, but without that picture for that puzzle, the cell can sometimes put it back together wrong. And so if it were to put the viral genome back together wrong, it could prevent it from basically leaving that dormant state. And for HIV, when you leave that dormant state, you progress from HIV to AIDS. So it can certainly, it has potential, and there's discussion about using it in that way. Using CRISPR as a way of attacking viruses is challenging, because CRISPR targets double-stranded DNA. That's when you've got two of those complementary strands of DNA together. And as we've really learned in the news today, there are a lot of different kinds of viruses out there in the world. And not all of them use a genome that is compatible with CRISPR technology. So I'm sure everyone's heard of coronavirus. Coronavirus is not a DNA-based virus. So CRISPR wouldn't be particularly successful in targeting and damaging coronavirus. But I think that class of virus where it inserts its own genome into the genome of a person, there's a great possibility there where we could use CRISPR technology to, after the fact, so not a vaccine that prevents you from getting the infection, but after you've gotten the infection, and after, say, a patient has contracted AIDS, that we can destroy the AIDS virus that's dormant in those cells and prevent that reactivation at a later date. But again, I do want to caution everything that this isn't something that's going to be happening tomorrow, or this isn't going to be something that's happening in a year. This is an idea that a scientist in the world has conceived, and there's a number of steps that have to go before we can get to the idea of using CRISPR as a treatment for viruses. But I think it's an exciting possibility, and it wasn't something that before Dr. Dowden and Dr. Carpentier developed this technology that we were ever thinking, well, I wouldn't say we would never think of it, but we just didn't know how to do it up till that time.
Find the idea of using CRISPR to remove HIV from cells to be really, really intriguing. But getting all of the HIV reservoir out of the body is a huge, huge challenge. And the only way that it's so far been done to date is through that sort of bone marrow donation kind of process that you were talking about. So you probably have to do something similar. You'd probably have to do the wiping out of the irradiation of bone marrow, and then engineer engineer bone marrow to be possibly resistant to HIV, and then also target any latent virus in there to get rid of that viral genome. So it's a really complex process.
Yep. Just like you said, getting rid of that population of virus cells, because you don't want to basically correct HIV by destroying the information in the genome in a cell and then just have it reinfected again by that population of virus that's swimming around in the bloodstream. So yes, exactly. It's a very complicated issue. But again, I feel like this technology is just a glimmer of hope for some of these conditions that we have to hold on to for a little bit longer before we can really see the fruits of what these discoveries are reaping.
Coming back to the difficulty of finding the hepatitis C virus, a question about what are the characteristics of the virus and infection that hinder the research process?
So hepatitis C virus, the discovery of the virus had a few challenges, and a lot of it had to do with the way that it's worked with in the lab. One of the big challenges is that the virus is present in low quantities in serum and plasma, which is what was being studied by many groups at the time. Another issue is that the virus does not replicate well in tissue culture. So tissue culture is a technique that's used in the lab where you can grow eukaryotic cells and then add things to them to study them or put them in different situations and look at how cells change, or you can grow virus in them. There's lots of different ways that tissue culture can be used. And there was no system for growing up hepatitis C in tissue culture, so that really hindered the study of it as well. And the only animal host other than humans for hepatitis C are chimpanzees. So these are highly intelligent primates that we want to limit the use of them in a laboratory context. And so and then there's also the situation of having all of these different quasi-species, because they're dealing with an RNA virus that has poor proofreading ability. And so there's all these different genetic sequences that you have to deal with to try to find some that are effective, because some of those are going to be defective. So there's all these different pieces and characteristics of that virus that made it very tricky from a research perspective.
Andre, I have another question for you, and this one I really have to read quite carefully, because it really moves into an area that I'm sure only you can answer this question. If a black hole is a bend-in of the 3D space-time fabric, would the collapse be going into a fourth dimension? And do we have any evidence of a fourth dimension?
Right. So first of all, fourth dimension is already here. It's called time. So and it is fully full class citizen in the space-time game. It is very different from spatial dimensions in the sense that we all seem to be traveling in time in one direction, and that's one of the great mysteries in physics. It's so great mystery that a lot of people just simply don't even ask this question, why is the time like that? But anyway, that's one of the things you have to worry about when you talk about the things like creation events, like Big Bang, or this end of the world events, for example, when you fall into the black hole and you're about to get squished, what is going on there? So the short answer is that yes, we do have four dimensions. It's three space dimensions, and the fourth one is time. And if you fall into the black hole, they funnily interchange in the roles as you go on. So typically you're used to a situation where you go left, you go left, you go right, you go right. In the black hole, once you fall in, no matter where you go, you're still going towards a smaller radius of it. Mm-hmm. So it's like, this one point that attracts everything, and no matter how, you try to avoid it, go even, in full thrusters, you're still going to end up there. So what happens next is the question is a black hole really like an Einstein gravity, or maybe it's something different? So that is really entertaining, also an academic question. But these things were academic 100 years ago, and now we have real black holes being measured and imaged and listened to when they collide. So people are very much aware that the physics might pop up with a few surprises when you take a look at the close vicinity of this object. So everybody is really excited about it.
Okay, I want to ask you a follow-up question because I was thinking about this when you were speaking, and the answer to one of your questions. Okay, we can see the outside of a black hole, but as we get closer and closer to the black hole, no matter which way it is that we're examining it, the black hole has stronger gravitational forces. I think I heard you right. So once you start to enter into that black hole to try and figure out what's going on there, how do you get that information back out there? Because you said that, you know, the gravitational force within inside the black hole is so intense. I'm thinking, how do we start to explore into that space, which is sucking everything into it?
Well, I mean, you can try plunking in and try publishing a paper out of there. That might not help. But of course, what people are on the lookout for is anything that comes out from the black holes, especially the notion of event horizon being a physical surface. Or just a membrane with one way, like, you know, you're not coming back out of there sort of sign. It's not going to be like, you know, stop sign floating in space saying don't go there. If you go there, like, you know, you're gone. Maybe, maybe not. So the possibilities of it, which are directly connected with the singularity theorems that Penrose proven. So if there is, if the matter behaves as far as gravity goes as a normal matter we have, so it's attractive, then you're pretty much going to end up crunched in one way or another. So you might be debating whether it's going to be the crunch of this sort or of that sort. It's very academic discussion if it's a crunch. But on the other hand, it might be much more interesting. And if the light rays that starts going together at one point can become repelled, then it might be possible to go and open up the interior of the black hole into another universe, or maybe even another place in our universe. So these things are known as wormholes. Once again, you might be, you know, imagining that like warp drives and wormholes are all like science fiction stuff. No, there is like loads and loads of papers on them. And people take this actually quite seriously. If you had a time machine, how would you do physics? Can you like really fly back into the time scene and change things like, for example, not enrolling yourself into the theoretical physics courses so your future career as a scientist goes bust, or like do other sort of version of the grandfather paradox. So, and this is science questions nowadays. So these are exact objects or other, like, you know, objects which be dominated by gravity. So in some sense, understanding black holes is understanding the building blocks of our reality. And they're really simple building blocks. They're just not very common to us because they're big and far away. And people hope to learn a lot by studying them.
Another question on the Crispr technology. Is this a kind of artificial evolution? How does that affect natural course of evolution, if any?
Oh, that's a really interesting question. Is this a kind of artificial evolution? For humans, we have really gotten around selective pressure in a lot of ways. So we're perhaps not evolving in the way that we used to be. People live a lot longer if they get a disease now. There's medical treatments that didn't exist 100 years ago that they would have died at that time, which they survive through now. You could argue that CRISPR could be an artificial form of evolution. You are changing the DNA of an individual or an organism, and you're improving it, certainly from our perspective, whenever we try and do that. Most of what we're talking about, though, with CRISPR technology, and let's limit the discussion to humans, is a correction to the normal as opposed to an improvement to something superior, if that sort of thing exists. Right? You know, that comes back to the analogy that we're not going to shoot lasers out of our eyes or climb buildings. We're not trying to evolve humans into superhumans. We're trying to fix the problems that exist in the genome. I wouldn't imagine that any scientist is going to randomly start CRISPR treating humans to see if any of their mutations improve some element of human life. That would be a very, very inappropriate way to use this technology and to perform science in general. So is it a different sort of artificial evolution? In plants And agriculture, one could argue, yes. But in humans, I would say it's probably, it's not, and I can't envision a way that it would be used like that in the future. Because that's not the way the technology. Mutations in evolution occur almost randomly in DNA, and that change leads to either a positive selection or a negative selection. And so CRISPR can't be used quite in as random a way as it would be needed to be used to allow for artificial evolution to occur. At least certainly not in my opinion. But maybe other scientists would disagree with me.
Dr. Van Houten, a question for you. Houghton and Alter had already proved some pretty strong evidence that NANBH was linked to the hepatitis C virus. So why was Rice's experiment so important?
Yeah, that's a really good question. And it comes down to, in part, to how we think about causation for infectious diseases and history of infectious diseases, as well as, so historically there was a bacteriologist called Robert Koch who developed a series of postulates around infectious diseases. And one of those postulates that's still used today is that the idea that a pure culture, or in this case genome, can cause the disease that is associated, that can cause the disease that we're talking about. And so from a historical perspective and from a causation perspective, his experiment was really critical in that sense of actively linking the hepatitis C genome to as the cause for non-A, non-B hepatitis, even though we had a lot of supporting evidence up to that point with the reactivity that Houghton showed from non-A, non-B hepatitis patients with the hepatitis C genome. There could have been other factors at play, and with that experiment of putting that genome into chimpanzees and seeing a resulting hepatitis, that really connected that, made that causation connection.
Mm-hmm. Thank you.
Another question for Andre. If nothing comes out of a black hole, then what would explain the soft hair?
Another great question. Somebody is an expert in the audience. Anyway, it's a little bit of a long story, but maybe I'll try to give a shorter answer in a slightly bigger context. So black hole is a really simple object. Apart from gravity, it has very little characteristics. So from looking outside of it, for the Schwarzschild black hole, you only have the mass of the entire stuff that, fell into it, not the history of it. And for the rotating black holes, like Kerr black hole solutions, you also have the spin of the black hole. So in some sense, they behave almost like elementary particles in the way they determined by, their properties, physical properties, are determined by just a handful of numbers. The question, of course, comes, how does this mesh with quantum physics? When you start throwing stuff in, it usually have quantum properties associated with this. For example, it might be in a pure state, it might have quantum numbers associated with this. You drop it into the black hole, and all of it seems to be forgotten. So this is the main sort of simple but show-stopping block between meshing the gravity and quantum physics. And it is still there. People don't know how to solve it, really. String theory was developed to address this sort of questions, but the big question is still being debated back and forth quite hotly. Where does the information go? When you drop stuff into the black hole, where does it go? Where does it get stored? One of the ideas along the software things was going back once again to the probably 70s, if I remember correctly. It was basically that there might be deformation of the information stored just outside of the black hole event horizon. So that's where this context of the question comes in. But I will sort of make it even a bigger puzzle, if you allow me. It's a question of where does this information goes after you flush it down the black hole, right? We have a good question about the goldfish analogy with the piping. It's somewhere down those pipes, but we have no idea what's inside the black hole. And it seems like this question might not be just academic one, but it's sort of key in understanding how the physical forces work. It is closely related to the quantumness problem. How do you make things small? Well, if you make things small in the laboratory, quantum mechanics usually steps in. But when you make really small black holes, will be the quantum objects, will be the fuzzy in their origin, like, will be the quantum things going on. Or maybe there is something else goes on and the answer is entirely different. People come up with a lot of crazy ideas in this thing, so it's still being actively debated. So I have no answer for it. That's a short form.
You know, you're already demonstrating that 2 hours isn't enough here, that there's a lot of questions, and we're technically like out of time right now, but we got one more question that we want to ask to Dr. Audus, and that is the CRISPR system, can it work on double-stranded viruses? Could they be used for treatment using CRISPR, or is that still a little too challenging? And of course, I'm going to give you another challenge and say, you got to give us a short answer.
Well, the short answer would be theoretically yes. If a virus genome is double-stranded and the CRISPR was present with the Cas9 and the sgRNA were available, it could be used to damage that virus. The practical answer is probably no, because you would need the CRISPR to be basically in the same cells that were being infected. And I think realistically, we're not at a place where we could do that right now. It's certainly an interesting question, but I will say it's both a yes and a no.
And I would just add that there's lots of other ways that we have to target viruses that are going to be way more effective.
I absolutely agree. Yeah.
Those are fantastic questions. It really shows how engaged our audience is and how interesting the research topics were. Thank you very much for your participation. Thank you, everyone. I wish we had more time to keep discussing, but now it's over.
So this has been a very inspiring evening. I felt like we were just really starting to get cranked up as we ran out of time. However, we have come to the end of our program, and I would like to thank Andre, Ninke, and Timothy, or Dr. Audus, Dr. Van Houten, and Dr. Frollo for presenting, being our presenters from Simon Fraser University for their inspiring research presentations. Thank you too to Karina for co-hosting this event with me. It's been a pleasure staging this event with the Faculty of Science at Simon Fraser University. Thank you as well to Dr. Paul Kench, the Dean of Science at Simon Fraser University, for this opportunity to engage the public in science research. And of course, thank you to everyone in the audience for joining us this evening and sending in your questions. We appreciated all of those questions that you and comments that you sent in, and I feel badly that we didn't get to them all because there were so many good ones. But thank you. And we hope to do this again next year, celebrate the next round of Nobel awardees. And remember, you can see the show later, because we are recording it. It will be available for those who are unable to attend, or that you can come back and revisit this online in the future. And in the meantime, please stay safe, keep healthy, and let's all keep the passion for science and research alive in all of us. Good night.